Today we are at UIO Plant Lab, the University of Oslo's infrastructure for climate-controlled plant growth. Plants have historically contributed to very important discoveries in the field of biology. For example, think of the first description of cells, or Mendel's contribution to genetics. Barbara McClintock, a famous botanist, also first described the use of transposons. Plants are the largest organism on Earth, but also the oldest. Have a look at this section of a 300-year-old tree, and the French Revolution is all the way there. In terms of developmental biology, plants are models for other types of plants, of course, but can also be extended to other organisms when it comes to fundamental processes in biology. For example, gene regulation, cell cycle, or signaling between cells. For such research, dedicated models are being used, and we are going to take a look. Hi, Jonathan. This is Riccarda Schimanski.
Nice to meet you. We'll show you the plant facility over there.
Great, looking forward. We have a GMO facility here that we are going to enter. Perfect.
So, Jonathan, what plant do you use as a model?
Here in this lab, we work with Arabidopsis thaliana as a model for all plants. It has all the benefits of a model organism. As you know, it's very small. You can grow it in large quantity. It has a short life cycle. It's small, has one of the smallest plant genomes, so it's easy to work genetically with. Like, we have a large collection of mutants, and it's easy to transform transgenic genes in there, or reported genes.
Okay, but can you use the knowledge from this plant also for other plants?
Yes. So what we learned in Arabidopsis thaliana so far, it's really the basic for now ongoing research in other plants, and it's very often a comparison to this what has been discovered in Arabidopsis thaliana at the moment.
Are there any challenges with working with plants?
Yeah, the big thing with this model, choosing this model organism, is that it's really simple and easy to work with. And this allows us to find new challenges also in molecular development and understanding this complex regulations, for example. Or we can also transfer this to other plants and see whether this is different in nature in other plants, for example.
So what's unique then about plants compared to other model organisms?
There are really a lot of differences between plants and other organisms, but for me, one of the first you notice is that's not moving around. It's quite stable, and you see this also during development. So it's restricted by cell walls, for example. So growth is more, it's different from other developmental model organisms, for example.
So what does the lab research on here, and what are your particular research interests?
So our main focus is on flower and seed development. So we're interested in the different tissues inside the seed, how they communicate with each other. We're also interested in how the contribution of the father and the mother via the genes regulate the seed development. This is called imprinting. In a wider perspective, we're also interested in how these developmental processes regulate or have an influence on speciation and hybridization between species.
So you're going to set up a cross now. What does that entail?
I'm going to cross these different plants to see how the seed development will look like. Therefore, first I have to remove all the flowers which are already pollinated, because it's a self-pollination. I will simply cut them away, just that they don't interfere. And then I take the oldest flower which is just about to open. And here I will then remove the anthers so that they don't pollinate these plants. Then I can take the other flower which provides the pollen and place it here on the stigma. And this can then go inside and pollinate the plant.
Great. So what are we seeing now?
So this is a Seedlings which contains the seeds, and we can place it here on this microscope slide, and then we have to use the needles to cut it open to actually be able to see the seeds.
How many seeds do each of these have normally?
It's roughly 50 up to 70, depending on the age of the flower and the sleek. But here you also see a mutant phenotype. So this plant has a defect which aborts early on during development.
So do you mean that the seed will not be able to give rise to a plant? Is that what you mean?
Yeah, this is dead. It's dried out. So here again you see nice healthy seeds and early abortion, late abortion. So there will be a really small embryo inside, while this one is already fully developed. Yeah. And here it's also easy to squeeze out the embryo. Yeah. So you see the root tip and one of the cotyledons on the left side. Here you can see the cotyledon, which are basically the seedling. And in the middle you see on the main meristem you can already see the first two leaves. And here on this bit older plant you see that you gain now leaf. And you also see on the leaf surface these large trichomes, which are huge cells, very, very strong, protected with a strong cell wall, and they do a lot of endoplication inside.
So the plant uses it as a protective?
I think it's really protecting this shoot apical meristem, which is inside in the center of this rosette. So after we sterilize the seeds, we can plant them on MS plates and seal them so that we don't get a fungus infection. Yes, we take the seeds and place them. Here they get the perfect condition for germination. After 6 days, we can see the seedlings. This is probably after 8 or 10 days that we have this, and then we can transfer them to soil.
So how old are the seedlings now?
They're roughly 10 days now. And then we can select the interesting plants, for example, the mutants or the reporter line we have selected on, and place them to soil.
With the roots.
And add a lot of water because they come from really humid condition inside this petri dish.
So we learned a lot of things today. Yes. Jonathan took us through a tour of the lab. We learned how to control plant growth.
And how to make crosses, how to fertilize the plants in the lab. And we also learned how powerful the system is, because it's really powerful in terms of genetics.
Yeah, Arabidopsis really seems like a model that's easy to modify and maintain. So the possibilities seem endless, and while this lab here focuses on fundamental research, I feel that the applications in the industry seem endless. So it was an inspiring visit.
Yes, it was.